we are going to start our new topic, which is actually necrosis. We have studied the definition of necrosis, but we are going to discuss it detail. We will discuss necrosis many aspects, from many aspects, like we will discuss, first of all, we will discuss definition of necrosis, and then we will try to understand mechanism of necrosis, how biochemical changes occurs in the cell, and cell will undergo necrosis. What are morphological changes associated with necrosis, and with many types of necrosis? What will be the consequences of necrosis? Ultimate consequence of necrosis is the death of the cell as well. So then at last we will summarize all these things. What is necrosis? Necrosis is actually a form of cell death in which cellular membrane fall apart. It means it's irreversible injury. Necrosis is actually death, and death is actually the last option when cell faces injury, severe injury, and loses his ability to come back in normal position or a normal state. Then cell move toward the elimination process, and this elimination is actually necrosis, and the necrosis is accidental death. It means cell is facing any undecided or any or undecided action or stress or injury. That's why cell moves toward the death. And actually, uh, if we define according to Robbins, is a form of cell death in which cellular membrane fall apart and cellular enzymes leak out and ultimately digest this cell. Now, please focus. There are many things in the definitions. Like, first of all, cell is severely damaged. Its plasma membrane, its cellular membrane is not even attached with the cell now after injury, and cellular enzymes, because there are many enzymes in the cytoplasm and there are many enzymes in the lysosomes. What are lysosomes? Lysosomes are cellular cell organelles, and they have a very harsh enzyme. They are very dangerous or variable pH, or with different pH enzymes. So when lysosomal membrane gets ruptured, or when lysosomes gets ruptured, then enzymes from lysosomes moves into the cytoplasm. When lysosomal enzymes moves into the cytoplasms, then what happened? Then these enzymes, due to their digestive nature, due to their dangerous nature, they start eating cytoplasmic cytoplasm and other organelle. It means they start eating the cells. Understand? It means when enzymes moves out from the lysosome due to the rupture of lysosomal membrane, then these lysosomal enzymes starts eating the uh, cytoplasm or other cellular organelles. This is actually irreversible process. When lysosomal digestion occurs, this is irreversible, and if this is irreversible, cell ultimately leads toward the death, and this is actually happens due to lysosomal enzymes. So when cellular membrane fall apart and cellular enzymes leak out and ultimately digest the cell, it means there are lysosomal enzymes which are present inside the lysosomes, and lysosome is an important organelle of the cell. And when lysosomals leak out of the lysosome, it digests the cell. And when it ultimately when lysosomal enzyme moves out of the cell, even they start digesting whole tissue even. So necrosis is not decided. First of all, necrosis is not programmed death. It actually happens due to leakage of enzymes, and enzymes leak due to any injury or due to rupture of membrane or cellular membrane. And ultimately, when enzymes moves out of the lysosomes or or move out of the cell, then it digests the cellular part, and then it digests the tissue ultimately. So this is actually necrosis. So in very short, in few words, necrosis is actually accidental cell death due to enzymatic leakage and the rupture of or and the loss of cellular due to the loss of cellular membrane. होता क्या है इसमें कि लाइजोसोमल इंजाइम्स अल्टीमेटली एक सेल है, सेल डाइजेस्ट होता है, सॉरी सेल जो है सेल डाइजेस्ट होता है विद lysosomal enzymes, and lysosomes are the most important organelle of every cell. Actually lysosome, why lysosomes have lysosomal enzymes? Because in normal condition lysosomal enzymes जो हैं वो हमारी बॉडी में सेल के अंदर किसी भी हार्मफुल मटेरियल के एंटर होने या किसी मटेरियल को तोड़ने के लिए यूज होते हैं. सो दे आर वेरी डेंजरस. दे कैन इवन डाइजेस्ट आवर साइटोप्लाज्म एंड अदर ऑर्गेनेल्स. सो व्हेन दे लीक, नेचुरली ये लाइसोसोम्स के अंदर ही रहते हैं. नॉर्मल सर कंडीशन में लाइसोसोम के अंदर ही रहते हैं, लेकिन बट व्हेन नेक्रोसिस अकर और एनी इंजरी अकर्स cellular membrane and the lysosomal membrane rupture, then these enzymes moves out of the lysosomes. When these enzymes moves out of the lysosomes, they digest the cells, they digest the cytoplasm of the cells, and then they move out of this cell. देन दे फर्स्ट ऑफ ऑल लाइजोसोम से बाहर आए, उन्होंने साइटोप्लाज्म डाइजेस्ट किया. देन उसके बाद क्या होता है, इवन सेल से बाहर आते हैं डाइजेशन करने के बाद भी, सेल को किल करने के बाद, तो देन वो टिश्यू को नेक्रोज करते हैं. सो दीज दो मोस्टली नेक्रोज अकर ड्यू टू सेलुलर एंजाइम एंड फ्यू अदर मैकेनिज्म्स आर आल्सो इन्वॉल्व. वी विल डिस्कस लेटर. So this is necrosis. Necrosis is actually not programmed cell death. It is an accidental cell death. It happens due to when reversible injury, when stimulus remain even after reversible injury. कहने का मतलब यह है कि अगर कोई टिश्यू के ऊपर continuous stimulus, injurious stimulus रह रहा है, it means due to continuous stimulus or injurious stimulus on any tissue, then tissue become or tissue or cell of the tissue cell attain its permanent change, attain its permanent damage. Then due to permanent damage and in or irreversible damage, cell lead toward the death process, and it's a necrosis. Okay, so actually there is another mechanism is involved in the with the necrosis. When is lysosomal enzyme or enzymes, when enzymes moves out from the this dead cell, then this enzyme, then our body's immune system detects the detect any wrong condition, or detect it this wrong condition, and our cells, our inflammatory cells, our special type of cells moves toward the site and removes this dead cells. उस dead cell को remove करती है जिससे enzymes लीक हो रहे हैं, तो हमारे immune system उस dead cells को remove करते हैं, की कोशिश करता है. then there is एक प्रोटेक्टिव मैकेनिज्म इन्वॉल्व होता है जिसको कहा जाता है इन्फ्लेमेशन. सो इन्फ्लेमेशन होती है. सो नेक्रोसिस में एंजाइम रिलीज होंगे तो हमारी बॉडी प्रोटेक्शन के लिए एसोसिएटेड टिश्यूज को, जितने भी साथ में टिश्यूज हैं उनकी हिफाजत के लिए, या वहाँ पर इन्फ्लेमेशन कोज करवा देगी हमारी बॉडी. सो इन्फ्लेमेशन इज प्रोटेक्टिव मैकेनिज्म, but we will discuss inflammation is our next chapter, because inflammation is not only protective mechanism. Basically it is protective mechanism, but due to inflammatory mediators or inflammatory chemicals also causes damage to the cell or tissue. Okay, so we will discuss inflammation, but now keep in mind necrosis has an associated event which is actually inflammation. Inflammation is actually a protective mechanism. It protects our tissue from enzymes which release during the necrosis process, or necrose, which release from the necrose cell. Okay, so this is enzymatic digestion. It means you should now I think we have well explained the necrosis. So necrosis is a form of cell death in which cellular membrane fall apart and cellular enzymes leak out and ultimately digest the cell. Okay, so this is the definition of process. So how necrosis happen? Necrosis is actually happens due to change in biochemical mechanism. Biochemical mechanisms of necrosis vary with different injurious stimulus. It means if injurious stimulus, it depends on the biochemical mechanism, depends on the type of stimulus or type of injurious stimulus, यानी कि किस टाइप की इंजरी है सेल के ऊपर, तो उस तरह से जो है नेक्रोसिस होगी. नाउ there are common or general necro mechanisms of necrosis, like failure of energy generation ATP due to hypoxia and mitochondrial damage. If we have already studied that hypoxic conditions can lead toward the death of the cell. Hypoxic condition can lead toward the necrosis of cell because of low oxygen supply. Our our cells mitochondria are unable to produce or compensate energy requirements due to deficiency of oxygen. Oxidative phosphorylation is unable to produce ATP due to the oxygen deficiency. So due to oxygen deficiency, less production of ATP occurs. And ATP, what is ATP? What ATP is an energy molecule. So if ATP production is low or less, then failure of energy generation occur. This is process actually failure of energy generation. So loss of ATP production or loss of or failure of energy generation due to hypoxia and other condition is mitochondrial damage. Usually in injured cell, if if suppose we have I have told you that during necrosis membranes gets ruptured, cellular membrane gets ruptured. It means if mitochondrial membrane gets ruptured, then mitochondrial damage occur. This is mitochondrial damage. So if hypoxia is there, hypoxia even if oxygen is sufficient but mitochondria is damaged, then फिर भी क्या होगा कि ऑक्सीजन जो है उसकी वो तो ठीक है, लेकिन माइटोकॉन्ड्रिया डैमेज है तो मशीन डैमेज है, और मशीन जब डैमेज है तो एटीपी प्रोड्यूस नहीं होगा. सो ड्यू टू माइटोकॉन्ड्रियल डैमेज failure of energy generation is also occurs. So hypoxia and mitochondrial damage ultimately lead towards the deficiency of the ATP, and deficiency of ATP is actually a deficiency of energy, or this is no if deficiency of energy occur inside the cell, it means cell is unable to perform its normal function due to low energy level. It means energy level maintenance is necessary for proper functioning and stability of structure of the cell. Okay, so if hypoxia occurs or mitochondrial damage occurs, then ATP production become low. So when ATP production become low, energy pump gets failure, then ultimately cell starts moving toward the necrosis or death. Damage to cellular membrane. Now another condition, another biochemical mechanism is the change in cellular membrane, change in we have damage to cellular membrane means plasma membrane. It means plasma membrane gets damaged due to why, how, due to it means listen carefully, please listen carefully. Our plasma membrane have many pumps, uh, ATP dependent pumps, energy dependent pumps, okay, and movement of ions occurs through these pumps, okay. So if movement of ions does not occur, if movement of ions do not occurs properly, then what happen? If movement of ion do not occur properly, then concentration of ion gets changed inside the cell and outside the cell. Then many types of changes occurs. So plasma membrane, if texture of plasma membrane due to low energy level or due to enzymatic digestion or due to injury, especially in necrosis, when plasma membrane gets injured due to any injurious stimuli, then due to rupture of plasma membrane, cytoplasm or interior of cell can leak out from the cell. And when lysosomal membrane damage occur, this is more severe thing, because when lysosomal membrane rupture occur, then lysosomal enzymes moves into the cell or moves out from the cell, and these lysosomal enzymes are very dangerous. They can digest the whole cell. So this is another way of necrosis. Now then, irreversible damage to lipid, protein and nucleic acid. As I have told you that leakage of lysosomal enzyme can digest lipids inside the cells, can digest protein or cytoskeleton of the cells, and it can also digest or damage the nucleic acid. It means DNA or RNA of the cell. So ultimately, energy from a failure of energy generation to damage through damage of nucleic acid is actually a biochemical mechanism of necrosis. Another thing is reactive formation of reactive oxygen species, ROS. What are ROS? We will discuss uh, uh, in our next lectures. Okay, what is ROS and reactive oxygen species, and how reactive oxygen species are involved in the killing of cell. Okay, now यहाँ तक जो है अभी आप नेक्रोसिस के मैकेनिज्म याद रखें जो आपको पढ़ाया. फर्स्ट ऑफ ऑल, failure of energy generation, ATP loss, and then rupture या damage of plasma membrane or lysosomal enzyme, and leakage of enzymes, leakage of lysosomal enzyme in the cell, and the and from cell into the tissue, and then irreversible damage to lipids, proteins and to the nucleic acid. And ये सारे के सारे सेल को अल्टीमेटली और टिश्यू को डैमेज करते हैं. Okay. What are morphological changes in necrosis? Morphological changes actually means change in the morphology and or change in the shape of the cell and change in the internal structure of the cell. Actually, there are two types of changes. Mostly, as a cell have two major parts like cytoplasmic area, and the another part is the nucleus. So cytoplasmic changes, first of all, what are cytoplasmic changes? Increased eosinophilia. It means on hematoxylin and eosin staining method, cytoplasm of necrotic cell takes more eosinophil and appears pink in color, or appears increased pinkish in color. So this is actually increased eosinophilia. We will discuss in detail later. So don't worry. Increased eosinophilia is actually a property when we stain the cell with hematoxylin and eosin stain, H and E stain, then cytoplasm of the necrotic cell take more eosin, and appears more pink in color. This is actually increased eosinophilia. And then another thing is, compared with the viable cells, the cell may have a glassy homogeneous appearance. This is another change in the cytoplasm. And another thing is more prominent myelin figures. Myelin figures accumulation occurs during the injury. In irreversible injury, very less amount of myelin figures can be seen. But in necrosis or irreversible injury, more prominent myelin figures, more prominent myelin figures can be or can observe in the cytoplasm in the necrotic cell. Then another thing is vacuolated cytoplasm. When enzymes have digested cytoplasmic organelles, it means I have told you that there are lysosomal enzymes which release from the lysosome, and these lysosomal enzyme digest the other organelles of the cell. So when enzyme digests cytoplasmic organelles, the cytoplasm becomes vacuolated. Means you can see vacuoles in the cytoplasm and appears mouth eaten. It likes जैसे कि कोई चीज़ किसी चीज़ को खा रही है, ठीक है, माउथ ईटन इस तरह से अपीयरेंस होती है सेल के अंदर वैक्यूल्स की. सो this is actually vacuolated cytoplasm. This is actually vacuolated cytoplasm and the mouth eaten appearance. But when we observe the cytoplasmic change under electron microscope, we will observe some more detail like discontinuities in cellular membrane, plasma membrane, and organelles membrane. It means membranes damage is another important feature of necrosis, and it can be seen in electron microscope easily. Dilation of mitochondria, it means due to abnormal oxygen concentration or damage of the mitochondrial membrane, dilation of mitochondria can be seen under electron microscope. Okay, because it's actually organelle, and the size of mitochondria is too small, we can only observe it under electron microscope. Then we can observe large intramitochondrial densities accumulated inside the mitochondria or dilated mitochondria. And due to these intracellular intramitochondrial densities, mitochondria lost their functions, and the ATP generation is also disturbed. Then disruption of lysosomes membrane. It means under electron microscope we can observe the membrane damage of lysosomes. So when membrane damage of lysosomes occur, enzyme leakage occurs in the cytoplasm. So these are cytoplasmic changes. Another thing is nuclear changes. Nuclear changes are of three types. So first of all, the we if we discuss they are actually dealing with the change in nucleic acid of the cell. Inside the nucleus, we have studied the cytoplasmic changes in our previous slide. Now we are going to discuss nuclear changes in necrotic cell. So what is pycnosis? Pycnosis is characterized by nuclear shrinkage and increased basophilia. You can observe in the central diagram like pycnosis, a central portion or nucleus of the nucleus inside the cell is dark blue. Okay, it is dark blue in color. So in pycnosis, nucleus appears more dark, more basophilic because of condensation of or shrunken of DNA or shrunken of genetic material. When shrunken or condensation of DNA or genetic material occurs, it appears more blue or dark blue or in more basophilic under hematoxin and eosin stain. Okay, so in pycnosis, there are two or three features. First of all, DNA condensation occur, shrinkage or DNA condensation occur, and dark shrunken mass appears or increased basophilia occurs. Okay. Then another thing is pycnotic nucleus. Then this is actually condensed nucleus. This condensed nucleus undergo nuclear fragmentation, and this is karyoraxis. Means pycnosis undergo karyoraxis, and this actually ये जो condensed nuclear material था ये fragments में टूट जाता है. In your right diagram you can see karyoraxis nuclear condensed nucleus breaks into fragments, and this is nuclear fragmentation. Okay, so when it appears in nuclear fragmentation, then obviously you can see fragments of nucleus, and but due to still they will appear basophilic, they will appear blue in color due to because of basophilic nature. Then another condition is karyolysis. Ultimately, nucleus after fragmentation move toward the karyolysis. Karyolysis is the lysis of genetic material, chromatin material dissolution due to action of DNases. And our DNA has our nucleus has enzyme like DNases, and RNases enzymes are enzymes. Many types of enzymes are present, like they can even break the nuclear material. So due to enzymatic action, nuclear material degeneration or dissolution or lysis of nuclear material occurs. And now basophilic now loss of basophilia occur means it means you can observe in your left hand diagram karyolysis occurs, and the dark blue color will is disappearing, and lysis of chromatin material occurs. This is actually happens due to action of some enzymes like DNases and RNases. These are actually nuclear changes occurs in the necrotic cells. Okay, so now we are moving toward the another thing. These are most important thing: karyolysis, pycnosis. I have told you that I will discuss discuss uh, eosinophilia, basophilia in my coming lecture. So don't worry about what is basophilia and what is eosinophilia and what is hemotoxin and eosin stain. You will understand in our coming lecture. But now at this time, please keep in mind what type of changes occurs in necrosis. There are two types of changes: cytoplasmic changes and nuclear changes. In cytoplasmic changes, you have studied the vacuolation, you have studied the more eosinophilia, less basophilia, and then dilation of plasma and cellular membrane. But in the nuclear changes, pycnosis, karyoraxis and karyolysis occurs. Okay, and increased initially in pycnosis basophilia increased, and then from pycnosis to karyolysis basophilia moves toward the high to low value. Okay, so then we will discuss the basophilia and we will discuss basophilia and eosinophilia and hemotoxin and eosin staining separate. So don't worry about these things. Please keep in mind changes, at least changes in the necrosis. Okay. There are few types of necrosis. First type of necrosis is coagulative necrosis, a name indicating that coagulation or preservation of architecture occurs in this type of necrosis. Usually, what happens in this type of necrosis? Usually, usually enzymes and protein with the when injurious stimulus act on the cell, then it damage the protein or structure of the cell. And when structure protein or structure of the cell gets damaged, then enzymes of organ enzyme of cell also gets damaged due to injurious stimulus. And when enzymes also get damaged, then there are no enzyme, there is no dissolution of architecture of organ occurs. When there is no architecture dissolution occur, or dilution or digestion occurs, then architecture remain preserved for a few days after death of whole organ, but architecture remain preserved. So this is actually unique feature of coagulative necrosis, that is architecture remain preserved for few days. It means cells are dead, but architecture of tissue or organ is actually preserved, is actually maintained for few days. And after few days leukocytes recruitment occurs, and leukocytes यानी कि more WBCs or more leukocytes moves toward the site of move toward the organ or necrosed tissue, and they release lysosomal enzyme. When newly recruited leukocytes released lysosomal enzyme, these lysosomal enzyme digest the preserved necrosed organ, and then this preserved then this architecturally maintained organ is digested by the enzymes of leukocytes, and then phagocytosis of this tissue occurs. So this is actually coagulative necrosis. Important feature is the preservation of tissue architecture for few days, at least several days after death of the cell. Okay, then more eosinophilia, but as cells are dead, there is no nucleus or karyolysis has been occurs, then or no basophilia will appear, more eosinophilia will appear. Then when tissue is maintained, when tissue is preserved, then more eosinophilic anucleated cell occur because of the damage of the nucleus. A nucleus will disappear, then eosinophilic anucleate cell will appear. And then another condition is another is thing is infarction. What is infarction? Infarction is actually area of ischemic necrosis, यानी कि infarction एक अलग से टॉपिक है. infarction क्या होता है? एरिया ऑफ ischemic necrosis होता है. ischemia यानी कि ब्लड सप्लाई की डेफिशिएंसी की वजह से किसी ऑर्गन में नेक्रोसिस हो जाना. तो ज़्यादातर कोगुलेटिव नेक्रोसिस उसी सूरत में देखी गई है कि जब ischemia हो जाए, ब्लड सप्लाई कम हो जाए, देन नेक्रोसिस होती है. एंड मोस्टली इट हैपन इन ऑल सॉलिड ऑर्गन्स एक्सेप्ट ब्रेन. ब्रेन में जो है कोगुलेटिव नेक्रोसिस नहीं होती. देयर इज अ लिक्विफेक्टिव नेक्रोसिस हैपन इन द ब्रेन. Now we are going toward another type of necrosis. So you can observe in diagram the kidney architecture of kidney, outer boundary of kidney, or appearance of kidney is preserved, is maintained, but all nephrons or kidney cells are dead, but a texture is maintained. This is actually coagulative necrosis. Okay. Uh, Another type of necrosis is liquefactive necrosis. Liquefactive necrosis is mostly occurs after bacterial and sometime in fungal infections. Usually, what happens in this type of necrosis, leukocytes means WBCs moves toward the site of infection, and they release enzymes. When they release enzyme digests the tissue or infected part, and this infected part after digestion by enzymes undergo necrosis. So rapid inflammatory cells accumulation. What are inflammatory cells? They are actually, most of them are leukocytes. So when they accumulated, they release enzymes. Enzymes liquefies the tissue. So actually digestion of tissue liquefy the tissue. Digestion of tissue actually appears in the form of liquefaction, means architecture of tissue does not remain preserved in coagulative necrosis. Tissue liquefaction occurs. Most of the tissue of central nervous system undergo liquefactive necrosis, and during and another thing is after phagos when leukocytes when enzymatic digestion of leucos and enzymatic digestion of tissue occurs, then phagocytosis occurs and removal of tissue take place from body. But you usually in acute infections, as in bacterial infections, the material is frequently appears creamy yellow. ठीक है, and frequently appears creamy yellow. It is known as pus. So, in acute bacterial infections, area of necrosis usually appears in the form of creamy yellow color, and this area is known as pus formation, and this area is known as pus. So, pus formation occurs in liquefactive necrosis. So, you can observe in diagram a brain liquefactive necrosis can be seen in brain. As I told you that in central nervous system liquefactive necrosis occurs. So in brain you can observe in diagram brain is undergoing liquefactive necrosis, and the yellow area of brain is showing the presence of pus as well. So this is actually liquefactive necrosis. Architecture does not remain preserved. Mostly happens as a result of bacterial infection due to the generation of enzymes released from the inflammatory cells. These enzymes liquefies the tissue, so architecture gets disturbed. It does not remain preserved for even for 2 to 3 days or for few days. It immediately so enzymes immediately liquefies the tissue. So this is actually liquefactive necrosis, and the pus formation as another unique feature of this type of necrosis. Another type of necrosis is gangrene necrosis. Sometimes we deals gangrene necrosis with coagulative and sometimes with liquefative necrosis, because gangrene is actually a formation is a formation of an area due to formation of necrosed area after loss of blood supply. Usually it occurs due to loss of blood supply to the limbs, especially lower limbs, यानी कि जो टांगे हैं, जो पाँव हैं, और ज़्यादातर ये पाँव में या टांगों में होता है. इसके अलावा जो है it can occur in the upper limbs as well, but usually most of time it occurs in lower limbs due to loss of blood supply. uh, uh, there are many other types of gangrene necrosis, many other reasons of formation of gangrene necrosis, but we are going toward the characteristics of gangrene necrosis. Tissue undergo coagulative necrosis as a lower limbs lower limb suffers with the loss of blood supply. Then it converts into necrosed area, and the architecture remain preserved. You can observe in left hand diagram. Dry gangrene is actually a architecture is preserved. Architecture is somehow preserved, but this is actually coagulative necrosis. But when this at necrosed area any bacterial infection occurs, then pus formation occurs. Then at necrosed area you can observe liquefaction as in right hand diagram. This liquefaction or presence of due to inflammatory cells or due to inflammatory cells enzyme area of liquefaction is actually occurs. This is actually wet gangrene or liquefactive necrosis. So dry gangrene is the area without the bacterial infection, necrosed area without in bacterial infection, but liquefactive is necrosis is actually area with lipids with sorry area with the presence of area with the bacterial infections, and it appears like liquid area, so like we have studied in liquefactive necrosis. So this type of necrosis is actually gangrenous necrosis, and gangrene formation is associated with many diseases, but major cause is actually loss of blood supply, insufficient blood supply. So this is actually dry gangrene and wet gangrene. So please keep in mind wet gangrene formation and dry gangrene formation. Liquefactive necrosis can be seen in gangrene when bacterial infection occurs, and the coagulative necrosis seen without bacterial infection. Another type of necrosis is caseous necrosis. Caseous necrosis mostly occurs in tuberculosis infections, TB infections. Mycobacterium tuberculosis cause tuberculosis in any part of body, but most of time in lungs. We can observe pulmonary TB. We can observe caseous necrosis. Uh, Usually on gross examination, you can see in diagram, in black color diagram, you have a lung necrosed lung tissue with yellow white color appearance, central portion. This area is actually necrosis, cheese like appearance. This is known as cheese like appearance. So caseous necrosis, tissue with caseous necrosis grossly appears white or yellow white color, and this is actually cheese like appearance. This is another distinction feature of caseous necrosis. Um Another thing is tissue architecture does not remain preserved. We have discussed in coagulative necrosis, architecture remain preserved, but you can observe in diagram, you can't say this is actually lung, because architecture is not preserved. This is actually necrosed tissue. Okay. Then another condition, there is usually in caseous necrosis, another condition can be seen is a granuloma formation. What is granuloma formation? In caseous necrosis, with caseous necrosis, usually many inflammatory cells can be seen, like collection of macrophages. Many macrophages fuse to form a central large necrosed area or central large area. This actually is the fusion of many inflammatory cells and and formation of specific area where epithelial cells can be seen, where many macrophages and lymphocytes can be seen. This area is known as a granuloma. We will discuss granuloma formation in our next chapter, inflammation in inflammation. But now keep in mind, caseous necrosis can be seen in mostly in granuloma formation, and most of time granuloma formation can be seen in TB, and caseous necrosis can be in TB infection. On microscopic examination, you can observe in right hand diagram a central area is actually showing caseous necrosis. Of course, uh, you can't identify this now, this clearly, because this is truly a new thing for you. So with the passage of time, you will be able to understand these things. So अभी तक के लिए सिर्फ इतना याद रखें कि फीचर्स क्या है. ज़्यादा टीबी में होता है, चीज लाइक अपीयरेंस होती है, टिश्यू आर्किटेक्चर प्रिजर्व नहीं रहता है, एंड ग्रेनुलोमा फॉर्मेशन कैन बी सीन विद द केसेस नेक्रोसिस मोस्टली, most of the time, and the collection of lysed macrophages usually can be seen in the microscopic examination with the central cases, with the central necrotic area. Okay, so this is actually this was actually cases necrosis. um, Not only tuberculosis, please keep in mind, most of the time it can be seen as TB infection, but it can occur in any type of bacterial infection. Okay, it was an example, like TB infection is example, but it can occur in any bacterial infection, but most of the time it can be seen in TB infections. Okay. Here is another type of necrosis, which is actually fat necrosis. Fat necrosis, when occurs, it occurs due to mostly due to action of lipase enzyme. Lipase enzyme released from the pancreas when during usually during acute pancreatitis. Acute pancreatitis is a condition, as a disease. Lipase enzyme release, lipase, a large amount of lipase enzyme release. So these lipase enzymes digest or destruct the fat, or when they cause destruction of fat, it undergoes fat necrosis. So usually there is a condition which is known as uh, calamitous abdominal emergency, known as acute pancreatitis, in which usually peritoneal, यानी कि जो हमारे एब्डोमिन की फैट है वो उसकी नेक्रोसिस होती है. तो when peritoneal fat undergo necrosis, then lipase enzyme digest or breaks the ester bond between triglycerides. When lipases breaks the ester bonds between triglycerides, then triglycerides breaks into fatty acids. These fatty acids binds with the calcium ion as present in blood. So these fatty acids, after binding with calcium ion, they show specific appearance on a microscopic examination. This is actually sponification. This is actually fat sponification, and it can only be seen in fat necrosis, as is there is destruction of fat. So this is actually fat necrosis. On histological examination, it means when we will observe the slide of the this fat necrosed tissue, then foci of necrosis contain shawdy outline of necrotic fat cells surrounded by basophilic calcium deposit. So when we observe the fat necrosed area, we can observe more basophilia surrounding due to deposition of calcium. So calcium ion will show more basophilia. Then you can see, or you can observe a diagram, a picture in a slide where pancreatic when where fat necrosis of pancreas is shown. So this is actually fat necrosis. I will discuss, revise these things with more detail easily. But now keep in mind, these are actually basic points in this fat necrosis. Okay, we are moving toward the another type, which is actually fibrinoid necrosis. Last but not least, another type of necrosis is fibrinoid necrosis. Fibrinoid necrosis is actually formation of fibrinoids. Can be seen in under a light microscope. Before, what are fibrinoids? Just leave it. I am going to discuss fibrinoid necrosis. Usually, what happens, our body have antibodies which bind with the antigens. So, and antigens antibody complexes are formed, and they get deposit on in the surface of blood vessels, in the internal lining or internal wall of the blood vessels. I'm going to repeat it. Antigen antibody complexes deposition in the wall of on the wall of blood vessel causes fibrinoid necrosis. Okay. It may occur in hypertension. Okay. So usually after this deposition, these deposited immune complexes and plasma proteins, it means our blood have the proteins, no? And plasma protein that leak into the wall of damaged vessel. When where these complexes get deposited, the blood vessel get damaged. So after at the site of damaged blood vessel, this damaged blood vessels usually produce a bright pink amorphous appearance. You can observe in diagram bright pink amorphous appearance. It means this is actually on H, and it can observe in H and E stain. So this bright pink amorphous appearance is actually is known as fibrinoid. Okay, now you can understand this. This bright pink amorphous appearance is known as fibrinoid, and So that's why this necrosis is known as fibrinoid necrosis. This is actually immunological. This fibrinoid necrosis occurs after immunological or immune reactions. Okay, there is another disease is polyarthritis nodosa, arthritis nodosa, in which you can observe a fibrinoid necrosis. I will revise all these things very soon with you guys, so don't worry about this first lecture. Uh, Try to listen again and again this lecture, and you will and please study it from book as well, Robbins. Okay, so it will be easy for you guys. Thank you so much. This is it, all these are all types of necrosis.